Good evening, ma'am.
Good evening, sir. You can start now.
Yes, ma'am. Thank you so much. Good evening, my dear students. You are all warm welcome to the biological session. Today, we are going to discuss about the first transgenic primate. There are so many examples already we have studied. The transgenic animals. Okay. And then transgenic fish. And next, we will pass on to the gene clue. Before that, we have to remind. Okay, I will remind you. So, what are the things already we discussed? Is small glands. Okay. The first transgenic primate. Okay. Dr. Gerald Shatton created the first genetically modified rhesus monkey called Andy. Already we discussed the rhesus monkey. Already we studied in the blood grouping, isn't it? Idea. Blood group that RH positive, RH negative. How it will be identified with the help of the rhesus monkey anti-serum. Okay, you can easily find out whether blood group containing positive or negative. Likewise, the first transgenic primates also discovered by the scientist called Gerald Shatton, a rhesus monkey called ANDI. For inserted DNA spelled backwards on October 2, 2000. So don't forget, my dear students, this is very, very important. First primate name is called as Andy. In which year? October 2nd and 2000. How it will be created the anti? So the anti containing jellyfish gene. Already you know what, but jellyfish is one of the aquatic animal, isn't it? It has the plasmids. The specific gene is responsible for the fluorescent color during the dark time. It will be blooming. That gene is isolated from the jellyfish and incorporated with the rhesus monkey. So as a result, what will happen? That glows green in the dark. Which one? The rhesus monkey also glows green in the dark. So see the concept. This is the main concept of the transgenic primate, particularly the rhesus monkey. See, this is the important things. Already saw it. This is the first GM primate. It may be possible in the next human being, like that they have stated the statement. See how it will be acted, how it will be. So you must isolate the, okay, we should isolate the, which one, you know, plasmid from the jellyfish, identify the fluorescent color responsible gene. Then you know about that. You have to make a cut with the help of the polyandromic sequences, with the help of the DNA construction, endonucleases. After that, it is also transferred to the rhesus monkey gene, and also incorporated with the help of the enzyme is called as a ligase, DNA ligase. After the complete recombination of these two genes, and also it is introduced into the host cell is called as E. coli or culture medium. After that, it will be again inserted to the monkey. So the jellyfish gloom at the night. Similar to the properties also transfer from where to where? Gene transformation from the jellyfish to monkey. So the monkey is also glues on night. See the next slide, you can easily identify. This is the slide. See that. So normal monkey look like this one. So normal monkey look like this one. So when it's dark night, dark, what happened now? It's a look like this is the structures. The glooming. So fluorescent colors will be appeared. So this gene is responsible for, what is that? recombinant biotechnology only it's possible. So there is an important one, my dear student. This was done by the name of the scientist already we discussed. Okay, so likewise, there are so many animals have been identified, newly invented, and so on. So, okay, so just go and check it in your global, Google. You will get the ideas. Different types of animals, different GMO vegetables, GMO animals, and also other things. Also, you can easily identify it with the name as well as the photos also, or pictures also. Okay, go through my ideas. We'll pass on to the next slide is transgenic fish also. How it we get fish? Already you know that fish is one of the aquatic animals. Okay, it only used for the food, isn't it? Food and egg, that things. How it will be transfer the genes from one animal to fish? So transgenic fish is a super fish of many species such as common carp, catfish, salmon, goldfish have been produced. They contain human growth hormone gene because these type of animals, they will contain human growth hormone genes. So it can increase their size of the fish. So what happened? They attain a size twice of that so by non-transgenic fish compared to the non-transgenic fish. Transgenic fish higher than that of the normal one. How it will be? Because the presence of the human growth hormone gene present in the fish. You have to isolate the particular human growth promoting hormone genes and also incorporate with the normal fish. You will get the result up to size twice compared to the normal fish. See that next one, it will be better for your understanding. See, this is the ozone pod. Here I have chosen a different types. So ozone pod is one of the species. Another one is, see here, Chinook salmon. So these are the two things is there. Here, the Chinook salmon, it has a growth hormone gene. So this growth hormone gene is also isolated from the Chinook salmon cell tissue or whatever it may be. You can isolate the nucleus from that one. You have to isolate the growth hormone gene from that one. And also there is a promoter is also, it is incorporated or hybridization with the help of the enzymes. After that, what happened, automatically it will be introduced into the another new one. So as a result, genetically modified fish you will obtain because these two combinations present in the single gene. So here the size is very big. At the same time, it is also a ozone protein. So as a result, what happened? A small fish, see here the small size, here the large size. When we are compared to these things, so you will get the large size. Okay, that is our product. That is our aim to produce. That is very, very important, my dear students. See this one. So this is the normal one. See this is the normal. This is the transgenic. So it is twice, not twice than that of the normal one. It is more than that one, 5-10 fold, isn't it? Growth of the fish. Next, we'll pass on the next one is transgenic mice. Okay, already know a little bit. It's one of the mammals. The transgenic mice, they have been developed for use to testing in the safety of vaccine before they are used on human. The mice is very, very important role in the biotechnology and therapeutic engineering and also medicinal biotechnology. That is very, very important, my dear students. So once you are introducing a new or invented the new vaccine, they will not introduced to the directly into the humans. There are so many, what is the screening process also done? Screening process. Okay, that is very, very important, my dear students. So after the screening process, it will also transfer to the, first they should check in the animals, particularly rat, mice, rabbit, likewise. After that, it may be chimpanzee. Then it will enter to the test testers. So many human beings also act as testers. Then it has many slat is there. After that, come to the conclusion, it does not produce any effect on human beings, means it will be introduced into the market. That is very, very important, my dear students. Particularly, example, polio vaccine is one of the important things. The transgenic animals are also used for the chemical safety testing. That is very, very important. Transgenic animals, already told you, the mice or whatever it may be, rabbits or cow or buffalo or whatever, chimpanzee or rat, rabbit. So whatever it may be, they are all used for the chemical safety testing. Suppose if it is a negative result, what happened? So it will not continue. What are the difficulties? That it will be rectified and then it will be introduced into the market, or once again it tested. It should be tested. Transgenic mice represented human disease model for AIDS, diabetes, cancer, and immune functions also. Okay. So because they are all considered, it is known as mammals. It has a specific mammary glands. Okay. It gives offspring young one as a young one. Okay. That is very, very important. See, go to the next one is, see this one. So this is considered a donor. Okay, the donor cell is also undergoes the blastocyst. Already know about what is meant by blastocyst, and the blastomy is up to 18 to 32 cells. During this stage, the embryonic stem cells also produced from this one. They are isolated, these cells, and also transgene. Which one we are going to transgene? We have to cut the DNA molecule with the help of the molecular scissors, and also a selectable gene also introduced inside a transfection. After the transfections, also undergoes the recipient. Recipient is also already in our body. It is also one of the where you already isolated from the genes. So the same species also used for the recipient. And implant means already in our body that the egg is also implanted in the a female reproductive part. Already in our body, it is a testy baby. Already we discussed the testy baby. The fission of gametes occurs outside the test tubes. After that, what happened? It will be introduced in the female reproductive part, particularly the endometrium. After the fixation of the endometrium, what happened? Automatically, it will be continuously undergoes the cell division process, just like that. So after the recombination of two genes, so it will be introduced into the implantation process with the mother. So the mother yield so both the gene present the single organism, that is called as what is a transgenic founder. Transgenic founder. That is very, very important, my dear student. After that, what happened? It will be produced again and again, means you will get so many transgenic mice. Okay. See, this is the important things. See the next one, you can understand. These are the two types of mice. One is a black one, another one is called white. Some of them are having the guinea beak. It's a white in color, but the eye is red in colors. These are all considered, it is known as transgenic animals. Okay. So the part 2, it will be finished up to this level, my dear students. See that one? The question part will enter into the next one. The polypeptide of human insulin are linked together by, okay, so linked together by which one? Phosphodiester bond, covalent bond, disulfide bridge, and hydrogen bond. What is the correct answer, my dear? Yeah, early about. Okay. Suppose take an example, say any one of the DNA molecule, insulin molecule. Take an example, say any one of the insulin molecule. There is a 3 chain is a chain. Okay, another one is B chain, and then C chain. So these are the 3 part is the C chain, as well as the B chain, as well as the A chain. Okay. Here, how many amino acids there? Already you know about the A, it contains 21 amino acids. Already we discussed 21 amino acid. Here 30 amino acids, and also here also 30 amino acids. Total how much? 81. 81. Just to calculate it, you will get it the amino acid sequence. So here the one end is connected with the what is that? Amino group, amino group because it is made up of amino acids, isn't it? Amino group. And the other end is also carboxylic end, isn't it? COOH minus. Here there are three parts, 1 and 2 and 3. You have to split into this one chain is called as A chain. This one is called as B chain. This one is called as C chain. So already given here. So in between A chain and B chain, E chain and a B chain. Okay, what happened? The structure will be like this. This is the A, this is the B, like that. The in between there is a bridge. That is called as, what is that? Disulfide bridge. Don't forget, my dear student, disulfide. Okay, SH bridge, sulfhydryl. So you will get the 2 SH. So that's why it is called as a disulfide bridge. Disulfide means 2 sulfur hydryl compound is there. Likewise, the same is there. Then only they maintain their structures. Another one is there is a extra peptide chain is also there. Extra, suppose if it is a extra C peptide present in the end, C peptide. Okay, it is nothing but the extension of the amino acid sequence, peptide. Okay, C peptide. If it is C peptide present in it, that insulin is called, this is the structure of insulin. Already you know, but isn't it? Insulin. Insulin, it's contain, what is that? Insulin, it contain, what is that? 3 chain. Insulin contain 3 chain, A chain, B chain, C chain. Corresponding amino acid is there, and connected with the disulfide bonds also there. Okay, suppose if it has an extension like that C peptide is there, it is considered as a pro-insulin. Pro-insulin, C peptide present in it, it is considered as a pro-insulin. Pro-insulin means, what is that meaning? It is nothing but the primitive nature. It is nothing but the primitive nature, pro-insulin. The C peptide is absent, it is considered a mature insulin, mature. Okay, only it depends upon the presence and absence of C peptide extension only, mature insulin. Okay, why it is called as mature insulin? The C peptide is absent, that is considered as a, what is that? Mature insulin. It acts as a human insulin. Okay, that is very, very important. This is also a very, very important, my dear students. Don't forget. So they may ask the question in this part, important one. And second one is, see that one. So this is the structure. The polypeptide of human insulin are linked together by a phosphodiester bond and covalent bond, a disulfide bridge. This is the correct one. It is not a wrong one. This is the correct one. So disulfide bridge. This is not a correct one. Okay, so already discussed. Next question is, the human insulin is being commercially produced from where? The human insulin is being commercially produced from the E. coli. Already you know about, there are so many genetic engineering also done by the E. coli. Mycobacterium tuberculosis, it is nothing but the mycobacterium. It is a TP producing microorganisms, particularly rhizobium used for which one, only the nitrogen fixation. Saccharomyces cerevisiae, nothing but the yeast molecule. It will not produce a human insulin. Rhizobium, it will not enter into the insulin production. Mycobacterium, it will not introduce. The E. coli already know very well, but it's most of the cases genetic engineering done by the, which one, E. coli only. E. coli is the very, very important, my dear students. E. coli is the correct example. So this one is the correct one. This one is the correct one. So here you just note it down in this category. See that things also we will discuss. So then you will understand. Okay. So they same things. Human insulin products. So after this one means, what is that? Okay. After this, see I will clear it, all those things, then only it will be better. See, so already in our board, this is the human cell. Okay, the particular gene is responsible for the insulin production, isn't it? Insulin production, insulin. Okay, insulin producing genes. Okay, so this portion, completely note it down, this portion. Okay, in this portion. So another one already in our board, that it's another alien species is also there. So alien species is containing another genes. Okay, these genes. Okay, you have to split the molecules. Already we discussed this part, all those things already we discussed this part. Okay, this part. Okay, you have to make a cut, same process is also done. Okay, as a result, you will get it a small piece of the B sample. This one is considered the A strain as well as the B strain. The particular gene is responsible for the production of insulin. Particular gene is responsible for the non-insulin, like that. Okay, imagine here, just imagine this one, red color. Okay, that is very, very important. See here, see that? So that red color. Okay, red color in responsible for the, what is that? Okay, this is the gene for the insulin. Okay, after that you have to change it here. Okay, it will be fixed with the help of this one, or here you can change it because this gene is responsible for the insulin production, isn't it? It will be implemented with the B genes. So as a result, the result will be like this. Okay, the result will be like this. This is the gene. Okay, from this one already in our board. The next one is, so the entire genome of the another sample. Okay, so after finishing this one, okay, this is the gene for A gene. This one is B gene. Okay, so it will be entered into the, which one, you know, the coast is very, very important, my dear students. Without coast, there is no any implementation. So after that, the completion of the recombinant DNA technology finished, you know, after that it introduced into the coast, isn't it? The coast is called as, what is that? E. coli. Okay, don't forget, my dear, E. coli. E. coli is a vector. Okay, vector. It can be easily, what is that, reproducible and short period also. That result will obtain very short period. That is very, very important, my dear students. So inside what happened, you know, already, you know, see, this is the structure of the genome. It will be introduced inside with the help of the microporation, gel electrophoresis, gene gun, or whatever it may be. You can imagine. So which things is that? It will enter inside, isn't it? This part is enter inside with the help of the gene gun or whatever it may be. So here, what happened? You know, the host also produce the two genes. These two is present in the single form, isn't it? So you will get the gene like this. So many copies. Okay, so this is called as, which one, you know? Act as a human insulin. Act as a human insulin. That's why it is called as humulin. Okay, humulin, human insulin. Humulin is also produced within the coast. Okay, so within the coast, you will get the copy of. Okay, that is the version. This entire structure is also done in vitro. Humulin is nothing but the insulin secreted by the laboratory in vitro with the help of the E. coli as a vector. So here you must note it down, my dear student. This is very, very important. Okay, see the next things. See, this is the correct. Okay, E. coli. And the third one is, see that third questions. Just note it down, my dear. Okay. The transgenic Bt cotton, Bt, Bacillus thuringiensis cotton. So the resistance against, don't forget, my dear students, it's a specific questions. Transgenic Bt cotton, Bacillus thuringiensis cotton. So the resistant against roundworm, no, bullworm, or otherwise called as bollworm. It's a correct one. Okay, it's a direct questions. Okay, but see here the Bt and boll. Just imagine like this. Okay, Bt as a imagine. So this is the correct one. And pinworm, it's not coming here. And all of these, it is not coming. So this 3 is the, you have to find out the answer, Bt cotton. So you must find out the answer. Bt cotton is nothing but the BO. See here, the first letter B and CO is the second letter O. Wool, warm is the correct one. Okay. So go to the next one. These questions is very direct question only. The name of the nematode that infect the root of tobacco plant. Last class we were discussed. Bacillus thuringiensis. It is nothing but the BT. It is not found in the tobacco. Agrobacterium tumefacium, tumor-inducing things. It is not a nematode. Melidogyne incognita. So it's a correct one. See that these three only. These two are bacteria. It is not comes under it is nematode. So the Melidogyne incognita is the correct one. So this is the answer. Okay, this is the answer. So this is the correct one. So you have to select answer. It's exactly which one is bacteria, which one is a fungi, which one is algae, which one is the not related to that one. So you have to find out that one. Then only you can easily answer it. Okay. The next one. Last question. Match the following. This is very, very important, my dear students. Very, very important. Suppose if you observe the class, you can easily identify the answers. Lysostapin. The lysostapin is very, very important. Where, the transgenic, okay, the milk producing, the prevent mastitis. So in cows, the mastitis in cows is the correct one. Polygalactorinase, you note it down. This is very, very important, my dear student. Polygalactorinase is nothing but the tomato cell wall is made up of, what is that? Layer is called as polygalactose. It can be easily prevented. Which tomato means the tomato name is called as flavor saver. The polygalactorinase, flavor saver. So correct one. And human lactoalbumin. Don't forget, my dear. Lactoalbumin is nothing but the one of the which one cow. So the molecular forming goat comes here. So this is also already answer given. Answer we have chosen. The lactoalbumin rosei. Rosei is one of the cow. It contains the milk. Which one the human lactoalbumin. And which one is the last one? Already about the molecular forming in goat is the last one. It is a match with the spider-like proteins. Spider-like proteins. So answer is the first one. Lysostapin is the third one. Where there is a third one is coming. Fourth one is the third one. Polygalactorinase, it comes with flavor saver. So the fourth one. The human lactoalbumin is the second one. Rosy. And then spider-like protein is one. So this question is very, very important, my dear students. This is a very, very important part. Don't forget, my dear students. Okay. So this is the important one. Okay. So the part 2 is finished, my dear students. Part 2 is finished. We'll pass on to the next one is part 3. Okay. Part 3. So in this part, what we are going to discuss, a human cloning as well as the cloning, cell cloning. That things we are going to discuss was in this part. What is by cell cloning, my dear students? Cell cloning, how it will be exact copy of animals or plants. It can be produced from the single cell or single cell of the plant or animal. It's considered as a cloning. See the first introduction, cell cloning. The cell cloning is based on the property of totipotency. What is meaning of totipotency? Totipotency is nothing but any one of the cell, it can produce their entire organisms, the entire animal. That condition is called as what is the totipotency. It is mostly found in the in the plant. So particularly you can take an example of tissue culture. Any one of the plant part. Take an example, so root, shoot, flower, and also anthers, or any one. It can be introduced into the growth medium or nitty medium. It grows a entire plant. Okay, so that is very, very important, my dear students. So another one is the pluripotency. The property of totipotency in animals, a plant. A pluripotency in animals. Okay, that is very, very important, my dear student. Cell cloning is based on the property of totipotency in plant, pluripotency in animals. So it is very, very important. So totipotency is the potential ability of the plant cell to grow into a complete plant. Okay, how it be? Already we discussed this part also, my dear students. Okay, don't forget. So this is also a very, very important, my dear. Okay, take an examples of any plant. Take an examples, any plant. Take an examples. Okay. So this is considered a plant, isn't it? Okay, take an examples. Here the upper part you know about and lower part already you know about. Upper part is the which one you know? You know about upper part. This one is the lower part. So this is considered a shoot system. Okay, and then this one is considered a root system, isn't it? Root system. In between there is a flower is also there. This is also a very, very important, my dear. So this is a very, very important. This is the important point. Okay, this is also another important. Okay, so this is the flower. Okay, this part considered as a flower. Okay, the tissue culture means what does that mean? So you have to take the plant cells. It may be a shoot tip or a root tip or any other flower part or floral parts like the anther, calyx and corolla, androecium and gynoecium. So you can take that one and grow into a culture medium, a specific, already you know about tissue culture. Maximum they were used the, which one you know, test tubes. Okay, maximum they were used the test tubes. Okay, and sometimes they were used as a conical flask. Okay, here it contains the nutritive substance. Some of them are, they will grow in the conical flask. Okay, this is considered a conical flask. Imagine. Okay, so here there is a nutritive medium. That is a nutritive medium is called as agar. Agar. Okay, agar. Okay, not only the agar and also other nutritive substances, phytochromates also included, and the elements, calcium, potassium, whatever it may be, whatever the requirement they need. So all those things also present in it. Here also that is a culture. Here after that preparation of this one, what happened, you know, it is also undergo the solidification process. It is also a special process occurs in the laboratory. After that, you have to split the molecules. Split any part of the plant part, it may be replaced here. Equal to introduce in aseptically, all are aseptically introduced in this part. Okay, so it is also covered with the cotton block. Okay, cotton block. It's also covered with the cotton block. All are done aseptically. That is very, very important, my dear students. Here also there is a cotton block because the unwanted entry of the microorganism. It can be kept into the oven for culture. Okay, culture process. Okay, it can be placed inside the test tube. Also that conical flask also you have to place it inside. See this one. So this is the place. Okay, you have to place it inside. This is the oven. Okay, oven. So light is also you have to pass. Then only the plant growth will be occur. So as a result, after few days or 1 week or 2 to 4 weeks, what happened? You know that here there is a single cell is absorb the nutrition from this one agar medium, absorb the nutrients. So as a result, you will get the mass of cells. Single cell you already placed it inside the agar medium. So after few days, what happened? You know, you will get the mass of undifferentiated cell present inside this concept. Here, what is that? Undifferentiated. We cannot able to undifferentiated mass of cells produced. Okay, here also the undifferentiated mass of cells produced. Okay, this is called as what is it? callus. Okay, so callus. After that, it may be undergo the further subculture. Likewise, this one, same process also done. After that, you will get the pure plant, let's say. Once it will be grow, so final step is what is it? Organogenesis. That process is called as organogenesis. It is similar to that of the plant. It is similar to that of the plant, like this. It will be produced. Okay, so it will be produced like this. So many plantlets, say. Okay, so many plantlets also originated. Okay, so many plantlets. After the organogenesis, root and shoot formation takes place, it can be transferred to the, which one, land. It can be transferred. This plant is transferred to the land area. Okay, in the form of platelets, in the form of plantlets. Okay, plantlets. So this condition is called as, what is it? Already we discussed. That part is called as hardening. Okay, hardening. So these are all comes under it is known as tissue culture. Likewise, these are all plant related. These are all comes under it is known as plant related. Animals also, how it will be, so isolation of any one of the plant or animals. The some physiological changes occurs within the lab. After that, it will be introduced into the female reproductive organ. It will produce the exact copy of the animals, but it will be similar or dissimilar in nature. It entirely different. That is very, very important. Okay, see the next one. It's a cloning. That things we are going to discuss about in this part. Cell cloning is based on the properties of totipotency or pluripotency in animals. Totipotency is the potential ability of the plant cell to grow into the complete plant. Okay, these things now I showed, isn't it, my dear? Okay, as in the form of tissue culture. It contrasts pluripotency is the potential ability of the cell to develop any type of the cell in animal body. For instance, nerve cell, kidney cell, and heart cell. You can take any cell from the animals. It can be produced their organ or entire animals. Such condition is called as, such a process is called as, what is it? Such ability is called as pluripotency. Okay, that is very, very important, my dear students. So you have to note it down here. Two things is very, very important. One is called as totipotency, and also another one is called as pluripotency. Totipotency is nothing but the plant cell grow into a plant cell produced into a single cell produce the entire plant. Pluripotency means a single animal cell produce the organ or entire animals. Okay, so go to the next one. Virtually all plants are totipotency, but only fertilized egg and stem cell in the embryonic blastocyst are totipotency in animal. You should note it down. Very, very important. All the plants, you can take any part of the plant body. So plant cells, it has a totipotency compared to the animal cells. The plants are virtual. All plants are totipotent. You can take any one of the root tip or shoot tip, because it's contained the meristome, it continuously devolution, it will occur. That's why it has a specific in plant. Animals, what happens? Some specific cell only it will be grow. Fertilized egg. Okay, that is very, very important. Another one is called stem cells. Already you know a little about it. During the delivery time, you will get the stem cell. It is used for the future. Already it's one of the most medicinal miracle. Already you know what about. The embryonic blastocysts are the totipotency in animals. Moreover, in contrast to animals, there is no real distinction in plant between somatic cell and germ lining cells, because we can easily identify which cell is called a somatic cell, which cell is called a germ lining cell. We can easily identify in animals, but in plant we cannot able to identify, distinguish in plant. That is very, very important medicine. This is also this may also ask in the NEET questions in assessment reasoning and direct question. Okay, like this. The first one is plant cloning. Scientists have genetically engineered agronomically important crop plant. Rapid production can be achieved using actively dividing meristematic cells and tissue culture. Already told you. Okay, the plant, how it will be produced in short period, millions of plantlets. Okay, small plant. How will you? Means with the help of tissue culture only. Suppose any one of the plants already completely destroyed. There is no absence exist from the world. It can be grow with the help of the disease resistant, free disease resistant plants also we can obtain in this technique is called as what is the tissue culture, otherwise called as somatic cell culture. Don't forget, my dear student, this is very, very important. Don't forget. And animal cloning is more difficult than plant cloning. This is because animals lose their totipotency or reaching the gas loss stage of animal development. This is the difference between the plant cloning as well as the animal cloning. During the plant cloning, it can be easily, but animal cloning, it has some specific difficulties also there. Due to the totipotency on reaching the gas loss stage of the animal's development, it will be difficult. That is very, very important, students. Next one is the dolly sheep, the first cloned mammals. It's one already in our board, the worldwide, and also one of the miracles had happened. So the first mammals to be cloned was dolly the sheep. This is the first one. This was a breathed out achievement by Ian Wilmot and Keith Campbell colleagues at the Roslin Institute, Scotland in 1997. Don't forget, my dear students, 1997. Don't forget. This is very, very important. Here, maybe the question, which one is the first cloned mammal? They may ask the questions that is called as dolly sheep. One. Second one is the first cloned dolly sheep who produced, who achieved. Like that, they may ask the question. One is called as Ian Wilmot and Keith Campbell's. It's also another one. And third one is called as which institute? That is also asked. Sometimes it's also Roslin Institute. But and one more is there where it's present is Scotland. Which year? 1997. So here the 5 question is there in the single paragraph. Okay, don't forget, my dear student. It carries genetic material solely from Eves. That is, the dolly does not have father. Don't forget, student. This is very, very important. There is this also during the cloning methodology, particularly Ian Wilmot did. The dolly does not have a father. Only the mother cell it will produce the young one. How it will be possible? Okay, there is a special technique is there. See the genetic at the Roslin Institute removed the differentiated mammary cells. This is the important point. This is the important cell. Mammary cells from the udder of the 6-year-old sheep. 6-year-old sheep, the udder is the mammary gland is there. The mammary gland, there is a cell is that cell is isolated. The cell were grown in the tissue culture and then concentrated of the serum nutrition was substantially reduced for 5 days, causing them to pause at the beginning of the cell cycle. This is the process. This is the important process occurs within the laboratory. That is very, very important, my dear student. This is the important point. Which one? The cell were growing in a tissue culture methodology, tissue culture. Okay, and then the concentration of the serum nutrients was substantially reduced for the 5 days, causing them to pause that beginning of the cell cycle. So as a result, you will get the, what is that? Cell growth is started. See the next one. In parallel, the preparation of egg obtained from a Scottish black phase Eve were enucleated. Scottish black phase Eve or enucleated. Removal of the nucleus is condition is called as enucleated. Exit the nucleus. After that, the memory cell and egg cell were surgically combined. So combined, COMBINED, combined in the process called somatic cell nuclear transfer. Don't forget, my dear students, how it will be introduced. How it will be introduced. This is the process. This is the important process. Don't forget, my dear. Okay. So this is the important somatic cell nuclear transfer. Somatic cells. So sometimes they may ask the questions in the expansion aspects. Already know about the somatic cell nuclear transfer. The memory cell and the egg were fused to introduce the memory nucleus into the egg. This fused cell now had key power of see here. So first of all, you should note it down in this part. Okay. Take an examples. So first of all, you take an example. So 6 years Which one? Sheep. 6 years old sheep. Okay. SHEEP. Okay. Imagine like the small, okay, sheep. Okay. Okay. Small sheep is there. From the utter part means already in our body. It's nothing but the mammary gland. Okay. Utter part. Okay. Taking a cell. From here, they took a cell. That is very, very important. So this is the part. Okay. So inside what there is a nucleus is there. Okay. Don't forget. So there is a nucleus. Okay. This is a sample A. Okay. This is a A sample. Another one is already in our board, enucleated cell also. Enucleated means it is also taken from the another part. Okay. Another part. Okay. Another part. Okay. So this is considered here. Okay. So this is also here. Another part. Okay. Just a rough diagram only. Okay. Just imagine utter. So utter cell, otherwise called as what is a mammary gland cell, utter. This is the lower part, isn't it? So lower part cell is also isolated. This is a sample A. A B sample, what? Enucleated sample. Enucleated. What is that meaning? So completely removal of the center part. Only the without nucleus. Absence of nucleus only. There is no nucleus. This is called as enucleated. Okay. Enucleated. Okay. Now, what they did know, these two are connected. This nucleus is inserted to here. Don't forget here. What happened now? The nucleus is transferred from the utter to this one. Okay, this part. So now this cell, so this enucleated cell carrying the A cell nucleus. It carries the A cell nucleus, isn't it? This can be replaced into the female inside. Okay, inside. So as a result, what happened? No, it yields here the single parent only, single mother only. There is no which one father, single parent only, particularly parent only. So okay, single parent only, particularly mother only. Okay, here there is no which one father, mother only. But here what happened after the introduction of the nucleus? What happened? It may be replaced into the implantation into the female. As a result, after that you will get it the what is that? Two or whatever. How many samples you have to place? So it can be obtained. Okay, you will get it the 2 or 3, more than 3. Okay, so like that you will obtain. So this is the first one. This is the second. So this is the process of cell cloning method. Cell cloning. Okay, don't forget, my dear. So this is very, very important. See that. So that process is called a somatic cell nuclear transfer. Memory cell and eggs were fused to introduce the memory nucleus into the egg. This fused cell now had the power of totipotency. This was electrically stimulated to divide. This cell could now multiply and form an embryo or blastocyst. It undergoes the cell division process up to the 32 or 82 like that. So that cell division where there is when there is cell division occurs, that conditions already in our body, it's also produce the young one. So just similar to that of the human beings. Take an example, fusion of the male and female gamete within the female reproductive part. What happened? Automatically the fusion of gamete as a result formation of zygote. Zygote undergoes the cell division, that is called as meiosis cell division. Single cell divided into 2, 2 into 4, 4 into 8, like that up to 32. When the 32 cell comes, that condition is called as blastocyst. Okay, the same process also occurs as a result after the completion of the 6 months. What happened? See that. So 29 out of 2027 fused couplets. Don't forget couplets. Couplets. Don't forget, my dear students. Develop into embryos, which were then placed into the reproductive tract of the surrogate mother. Surrogate means which one is getting. Okay. So nowadays, you know about that. There are so many human beings also done this method only for the financial crisis. Surrogate mother. Okay. So a little over 5 months later, only on July 5th, 1996, one sheep gave birth to lamb named Dolly. The first clone generated from a fully differentiated animal cell. This is the very, very important. Dolly matured in an adult Eve and she was able to reproduce and old-fashioned way, producing a 6 lamb. He produced how many lambs is the 6 lamb were produced during this process. That process called as a, what is that? How many samples have taken? 227 fused, but 29 only. But from this 29, 6 only, you got the lamb. Remaining all are failure. Okay, see, this is the corresponding mammary cell donor from the udder. Taken cell, here enucleated cell, donor. Okay, so both are united. Okay, so as a result, you will get the lamb. Growth in culture and the implantation into the uterus of the third sheep and then embryo development. Okay, so here what happened, you know, the fusion of these two somatic cells, here the nucleus has taken, here the cell has taken. These two are not participating. It's also implemented into the third sheep. Okay, so this is the only one sheep. So who is the father of this one or mother of this? Mother only is there, no father. Okay, so this is a very, very important. See, the next one is also the story of Dolly, Scottish blackface, egg donor, the fin Dorset nuclear donor. The culture, the both are used to enucleated one. That means removal of the nucleus from this one, you will get this much. So here the nucleus you have to isolate. These two are interconnected inside. So A and B, you will get a single cell with the nucleus. So both the genes characters undergo the blastocyst and is also enter into the surrogate mother. Finally, you will get the Dolly lamb. This is the entire story of the Dolly. Okay, see that one. This Dolly established beyond all dispute the determination in animals is reversible, that which the right technique, the nucleus of the fully differentiated cell can be reprogrammed to be totipotent. Since she carried the nucleus from the fin Dorset Eve, Doll was a fin Dorset too. Dolly lived her entire life at the Roslin Institute. Edinburgh Dolly was allowed to breed with the Wells mountain ram that resulted in the birth of 6 offspring. Same is also done by the one other scientist called as what is a Wells mountain ram, Edinburgh Dolly. Okay, see this one. At age 5, Dolly began to develop complications in her joint and develop arthritis. So this is the problem, because immunity related things the scientists they didn't think. So Dolly survived for 6 years. This is very, very important, my dear students. 6 years. How many years they will be survived before she was euthanized because she was suffering from the arthritis and lungs problem. Don't forget, these things was not noted by the scientists. So on 14th February 2003, the 6-year-old Dolly was finally put to sleep. The life expectancy of Finn Dorset was about 12 years. How many years? 1997 to 2003, 6 years. Finn Dorset was about 12 years. See the difference. This is 6 years, this is 12 years. It has been speculated that his shortness lifespan is due to the use of somatic cell taken from the 6-year-old sheep. See, come to the conclusion. He come to the conclusion we should take the somatic cell from the 6-year-old sheep. That's why the lifespan is also very shortened. He come to the conclusion. Okay, so don't forget, my dear student, this is also a story. And table, list of the other cloned animals. What are the other cloned animals also done? How? You know, so these are the African toad. Kumlina is one of the mouse. Mira is a goat. Kupit and Diana is one of the sheep. Docom, pigs. And CC 2001, copycat, cloned cat, simply called as CC. Okay. Tetra rhesus monkey, first primate. No, as in Gaur, Bose, Gaurus. Nettie, Ditto, rhesus monkey. Charge, Charlie, cow, bull. Megan, Morgan, sheep. These are the name. Just note it down, my dear students. So these are also done by the scientist. That is very, very important, my dear. Next one is a very, very important, my dear students. This is important point, particularly in the bioremedy. Without bioremedy, there is no any living organism. We cannot be able to survive. The application of biotic agent like a microorganism in the correction and recovery of environmental damage is called as bioremediation. Suppose take an example of any one of the animals dead, what happened? Automatically the fungi, saprophytic also act upon it. As a result, what happened? The complex molecule is split into simple molecule. Then only the life cycle will be progress. Suppose if it is not possible, what will happen? All the dead animals present as it is, there is no any degradation process. What will happen? Okay. The biogeocycle is not progress. So the bioremedy is very, very important. See, the removal of the oil spill on a sea water by the help of bacteria is one example of bioremediation. Already, I told you. Okay. Particularly, which one is the, this is called as superbug. This name is called as, what is it? No, this name is called as, okay, superbug. Okay, superbug. Don't forget, my dear student. This is very, very important. Don't forget, my dear students, superbug. Which one is superbug? The Pseudomonas putita is a superbug. Why it is called a superbug? So oil spill in the ground, it can be easily destroyed. The simple molecule converted into, complex molecule converted into simple molecule. That means, see here, can easily identify petrol. Okay, otherwise called as crude oil. Crude oil is there. The crude oils, it is a hydrocarbon. It is made up of which one? Hydrocarbon, isn't it? Hydrocarbon. It's a complex hydrocarbon. You can tell as the complex hydrocarbon is there. The complex hydrocarbon, what happened? Spread on the ozone. Okay, ozone. What happened? Completely the aquatic animals suffered, isn't it? Aquatic animals or affected, isn't it? Affected. Animals are affected. This is very, very important. Once again, repeat for you. Crude oil is there. The crude oil made up of hydrocarbon, complex hydrocarbons such as petrol, kerosene, oil, tar, whatever it may be. Okay. So when it pour in the ocean, the aquatic animals are affected by the process. Okay. Aquatic animals are affected. How will we recover? So with the help of Pseudomonas putita, that is nothing but the superbug. G, superbug. Okay, don't forget, my dear students, superbug. Superbug, BUG. Okay, superbug. Which one? The Pseudomonas putita. When you are applying the superbug, what they will do? Already about the complex hydrocarbons all split into complex molecules. Complex molecules of carbon split into simple compound such as carbon, hydrogen, oxygen, nitrogen, sulfur, whatever it may be. Okay, so carbon, hydrogen, sulfur, nitrogen. Because carbon, hydrogen, sulfur, nitrogen, these are the, what is that? Component, isn't it? These are the component made up of, it united together form as a hydrocarbon, hydrogen, oxygen, carbon, isn't it? See this one. So as a result, aquatic animals are, what is that? Safe. That is called as bioremedy. So additions of bio, microorganism, and the environment comes up to the normal position. That is the application of biotic agent like a microorganism in the correction and recovery of environmental damage is called as bioremediations. Example Pseudomonas putida, it is also called as superbug. Okay, see that bioremediations. There are so many things is there. Okay, as a result, what happened? They are all are come to the atmosphere too. Atmosphere, some of them are atmosphere, some of them are water pollution or air pollution or land pollution, whatever it can be completely removed by the animals, microorganisms present in the soil as well as the water, completely recover the things, and also finally it will give you a good atmosphere, create the good atmosphere. That is very, very important, my dear students. Okay, bioremedy. So bioremediations, remaining things we will see the next classes, my dear students. Bioremedy and their types and ex situ and in situ and gene therapy. Okay, and basic gene therapy and types of gene therapy. These things we will see the next class, my dear students. Okay, thank you so much for the participation, and keep on practice. You will get the good result. Thank you. Bye. Take care. Good night. Thank you, ma'am.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, Ganna. Thank you, Ajay.